Today I will be talking about distorting DNA evidence through methods of math distraction. DNA evidence can be left at a crime scene and connect a person to that scene. Most often, a prosecutor wants the DNA in, and a defender wants the DNA out. Sometimes it goes the other way. Here, the prosecutor wants to connect the crime scene to the defendant, whereas the defender wants to disconnect that association between defendant and crime scene. The defender's approach is to attack the DNA proof. The prosecutor tries to maintain clarity about DNA evidence, whereas the defender tries to sow confusion. One can try to confuse a jury by persuading them that DNA is something that it's not, or introduce misleading statements if a judge allows it. The approach in this talk is distorting the DNA match statistic. Before we begin, let's review the meaning of a DNA match statistic. A DNA match statistic is a number that tells us how much more or less probable a match is between evidence and a defendant relative to a coincidence. This concept is familiar from simple DNA. Here is an example at one genetic locus. The random match probability, or RMP, is the chance that someone selected at random from the population has a particular genetic type, or genotype. The match statistic is 1 over the random match probability. That likelihood ratio, or LR, number is always greater than or equal to 1. As shown in the center of the slide, the match statistic is the probability of an evidence match divided by the probability of a coincidence match. With simple DNA, the probability of that match comparing very simple evidence and a person is 100%, which is the height of the blue bar shown on the right. The chance of a coincidence is shown in the brown bar. Suppose at this locus, for a particular allele pair genotype value, that rarity, or chance of coincidence, is 20%. Then the ratio of the blue bar to the brown bar of 100% to 20% gives a match statistic of 5. This number results from dividing 100 by 20. That was a match statistic at one locus. With multiplexed short tandem repeats, or STRs, scientists test 1 or 2 dozen different locations. Multiplying all those locus match numbers together, the final number can be much bigger than 5. But for one locus test, the slide shows the picture of a match statistic. There is not much confusion with single-source DNA. However, most DNA evidence today is a mixture of 2, 3, 4, 7, or 10 people who have commingled their DNA at a crime scene. The DNA item can be in a body cavity, on a touched surface, or on a handgun. This is what the data looks like. There aren't just 2 peaks, there are taller peaks and shorter peaks. Moving from left to right on the slide, boxes on the top of the peaks indicate genetic size variants called alleles. The height of a peak gives an indication of quantity, how much of each allele is present. Here, there are many peaks. Data like this are analyzed in the modern world by computer interpretation of the evidence. Computers can model how PCR behaves, how genotypes are added together to form a signal, and how various artifacts occur. Uncertainty can be modeled from the data. When computers interpret the data, they can consider every possible combination of genotypes. Richer modeling requires fewer subjective or arbitrary assumptions. All the data can be used without picking and choosing alleles, thereby eliminating human bias. The computer can then propose thousands or tens of thousands of different genotype combinations. Here, assuming 3 contributors. The figure shows a combination, in orange, of tall peaks from allele pair 15-18 in blue from genotype 1719, and in green at 1416. Adding these genotype contributors together forms a pattern. With this genotype proposal, the heights of the colored bars look similar to the data pattern. A better explanation confers more probability to each contributing genotype. That is how computers use all the data. They propose many possible explanations. Some proposals better explain the data, and some are worse. Ultimately, this consideration of many alternatives produces a probability distribution for an uncertain genotype. The genotype variable is uncertain because there are many possibilities, not just one. These genotypes are objectively generated, derived by examining all the data. The computer doesn't see the comparison reference during its examination of the data. After 3 people combine their DNA, police collect the mixture item, the lab produces data signals, and the computer separates the data signals into 3 genotypes. The computer derives genotypes from the data, 1 for each contributor at a tested location. With 3 contributors and 15 STR loci, there are 3 times 15, or 45, different genotype variables. Like most variables in science, genotypes have uncertainty, expressed here as probability. The slide shows one of those 45 genotypes for 1 contributor at 1 locus. On the horizontal x-axis, we see about 10 different allele pair possibilities: 1415, 1416, 1417, and so on. There were originally about 100 possible allele pairs at this location. Through Bayesian update, the data concentrated probability at the more likely contributor allele pairs. So most of the probability is now placed on these 10 allele pairs instead of being spread out across all 100 possibilities. The vertical axis is probability, also indicated as a percentage. This is what a genotype looks like when a contributor is separated from uncertain data. Probability represents genotype uncertainty, whether it arises from a DNA mixture, or kinship relations, or degraded DNA. The genotype is a probability distribution over allele pair values found before any match comparison is made. What is a match statistic? After a computer produces an evidence genotype, the question is how its allele pair probabilities, shown in blue, compare with those of a random person from a coincidental match, shown in brown. The same blue bars from the previous slide are now skinnier. The brown bars show the genotype probability for a random person. At this point, a comparison can be made with someone's genotype. That person could be a suspect, a victim, an elimination or contamination reference, and so on. The LR match statistic is the ratio of the probability of an evidence match divided by the probability of a coincidental match. When comparing with a person who has a 1417 genotype value, circled in red, we see that the ratio of the genotype probability is 24% in blue divided by 3% in brown. Note that the genotype probability is the same as the evidence match probability. So, at this one locus, the probability ratio is 24/3, which equals 8. That simple ratio gives the match statistic. It considers the evidence in blue and a random coincidence in brown for a particular person in red. Comparison can be made with anyone's genotype. At the 1617 genotype value, the brown bar is taller, so the chance of coincidence is greater than for a match. That ratio supports an exclusionary match statistic. when the probabilities of match and coincidence are equal, as we see at 1718, the result is neutral with a ratio of 1. We see inclusionary statistics at 1417, 1419, and other allele pairs. How can something this simple be made confusing? Well, it's been done to DNA evidence for over 20 years, and that's what this talk is about. Science gives a simple ratio, but court is an adversarial process where some people view their job as sowing confusion. How? Well, the general ploy is to distract the jury with irrelevant arithmetic that looks compelling. This can work in the courtroom on lawyers and juries who are not math types. I will present 3 scenarios. For each one, I'll show you the ploy, along with some testimony. The testimony comes from a 2016 Pennsylvania trial I consulted on. I'll also show how the ploy works, what's going underneath, and how a prosecutor can respond. This is a balanced presentation showing both how to distract and how to rebut. Ploy number 1, the highest probability. The 1719 allele pair, all the way on the right, has the highest probability at 28%, but the defendant's 1417 allele pair is less, at only 24%. If the defendant's allele pair doesn't have the highest probability, the ploy goes, it must be the wrong person. But that isn't how probability works. The probability of snow in Seattle may be low, but it happens sometimes. So how do we understand this ploy? Here is example testimony using that ploy. The testimony does not follow our pictures, but comes from a different case. The defense expert says there is an 8-11 allele pair at a locus, but it was only the 4th most probable genotype. "Explain what you're saying to us," asks the defender. The expert responds, "There were 3 genotypes other than the allele pair 8-11 that have been accounted for at a higher probability." And then the defender and expert go on to list other loci where the genotype value of the defendant didn't have the highest probability. The fallacy here is that we seek an LR that compares the strength of match between evidence and a particular person relative to coincidence. The 1719 allele pair on the right is not relevant to the match statistic. The real question is how the evidence, shown in blue, compares with the defendant's 1417 genotype, circled in red, relative to coincidence, shown in brown. The match statistic isn't about the biggest probability, it's about probability ratios. At a particular person's genotype, the probabilities of match to coincidence are compared. How might the opposing lawyer respond? In this case, after some sidebar discussion, the prosecutor said, "I am going to object to the relevance of this, unless they can bring some sort of expert opinion to bear on it. What's the significance?" The judge sustained the objection. Fallacy number 2, adding up all the other possibilities. This also isn't real math. Here's what's going on. Circled in red is our defendant with his 1417 allele pair, having a 24% probability. The distracting lawyer observes 100% minus that 24% is about 75%. And 75% is 3 times bigger than the defendant's 24%. Shown in green, adding up the probabilities of the other genotypes gives a number bigger than the one for the defendant. So the probability is higher that it's someone else, right? That arithmetic sounds pretty compelling, if you've never seen this ploy used before. To see what's going on, let's examine some defense expert testimony. The defender opens by asking, "Are these probabilities based on 100% or something else. The defense expert replies, at each location there must be another set of genotypes that have probabilities that sum up to the difference between 100% and whatever it is for one person. So, for that first CSF location, that 9% locus, there must be other genotypes that sum up to 91%. We have to sum up to 100%, so there are other possible genotypes. The defender asks, so we are talking about merely 9% out of 100%? That doesn't sound very convincing. Correct, responds his expert. Let's explain this fallacy. What we care about is what's relevant, shown on the left, the evidence to coincidence ratio for the defendant, not for anyone else, not for other people, not for other genotypes. That's not what a match statistic is. The other genotypes, those other probabilities in green, are simply not relevant to a valid likelihood ratio statistic. The relevant probability ratio view is up and down. It doesn't look sideways at other people. For a particular person, circled here in red, it's the ratio of the probability of a match after you've seen the evidence in blue divided by the coincidence before you've seen the evidence in brown. That's the legitimate match statistic. How to respond? There was no particular testimony on this given at trial. The ploy didn't seem terribly persuasive to anyone. Generally, the Rule 401 test for relevant evidence applies. Evidence is relevant if it has a tendency to make a fact more or less probable. The attempted ploy satisfies Part A, since it is indeed making something more or less probable, but it fails the second Part B. The irrelevant probabilities are not a fact of consequence in this trial and do not determine something about the DNA. The defender's added up probabilities are totally irrelevant to the DNA match statistic. A third ploy is that the match probability is small. It's only 24% at this locus, shown at the bottom of the slide, and also written at the top for the first locus. At a second locus, the match probability is only 9%. At the third locus, it's only 13%. When multiplying 15 numbers together, each less than 100%, the product is a small number. In the testimony, the defender asked, and did you come up with genotype probabilities after multiplying all these probabilities together? Yes, came his expert's reply. And what's your figure? Asked the defender. Roughly 1 in 3.5 billion, said the defense expert. That's the probability that the suspect matches the evidence. Seems really small, right? The distracted jury is thinking, the prosecutors were talking about these big DNA numbers, but only 1 in a billion chance of a match? Oh my, what's gone wrong with math today? Here is the fallacy. The match statistic is a ratio. The defense is totally ignoring the denominator. Multiplying the numbers, shown in blue, 24% times 9% times 12% and so on, will produce a small numerator, like 1 in a billion. But the rarity, the chance of a coincidence, shown in brown, is 3% times 1% times 4% and so on. This product gives a very small denominator, like 1 in a billion billion. And 1 in a billion divided by 1 in a billion billion is a billion. That is, small divided by very small is big. This arithmetic can blow a juror's mind, but that's what's going on. This misdirection can be very distracting. It focuses on a small numerator while intentionally leaving out the very small denominator. A valid match statistic requires both parts, numerator and denominator. The response came on redirect. The prosecutor asked, "What's the significance of multiplying these probabilities together?" I replied, "It doesn't have any, because it's just multiplying together the numerators, the top part of a fraction, for the probability of a match. The match statistic at each location is the probability of a match divided by the chance of coincidence, and that other equally important half of the calculation was left out." When someone offers a distracting DNA argument, even if seemingly relevant, the evidence can be excluded through Rule 403. The trial judge can balance an unsound argument's lack of probative value against the very real risk of confusing the issues or misleading the jury. Methods of math distraction are not relevant science and should not persuade anyone. By understanding misleading ploys for distorting DNA evidence, informed lawyers can neutralize deliberate confusion. For more information about valid DNA evidence, please visit us online. Thank you very much.